I'm Charlie Manning, Open Science Manager at the Focus Ultrasound Foundation, and I'm joined in moderating today's webinar by Frederic Padilla, our Director of Applied Research, who is also working on our data repositories. Today, we're going to talk about some of the policies and platforms that we have developed here at the Foundation to further open science and focus ultrasound. We're also pleased to be joined by speakers from two organizations which are doing much to advance openness and accessibility in our field and many others. And those are David Mellor from the Center for Open Science and Holly Smith and John McDunn from Project DataSphere.
Thank you, Charlie. So today's webinar will be composed of these few presentations, followed by a Q&A session. So please use the Q&A button on the webpage to submit your questions. Our first speaker will be David Mellor, the Director of Policy Initiative at the Center for Open Science. David leads the policy and incentive programs at the Center for Open Science, and is going to talk to us about this Center for Open Science, which is here in Charlottesville, and provide us an overview of what data transparency is and how pre-registration works. So David, the floor is yours.
Thank you so much for the invitation, and thank you for the introduction. I am sharing my screen now, so I think I just took over everyone's screen. I will assume that's working correctly until I hear otherwise. But yes, my name is David Mellor. I'm from the Director of Policy at the Center for Open Science. We are a mission-driven nonprofit to working towards increased reproducibility and transparency in scientific research. And I'm going to give a little bit of an overview of what motivates our work and how we focus on trying to achieve our mission. Information about our finances and support are available on our website, and I will provide a link to the slides also throughout the webinar. So I always like to start with a little bit of a description of a collective action problem that we see when thinking about and addressing increased transparency in academic research. So about 10 years ago, Anderson and her colleagues surveyed all recently funded NIH researchers and asked them to compare and contrast the Mertonian norms of how science perhaps should be conducted. And those are all about open sharing of materials, organized skepticism, and an overall focus on quality over quantity, and compare those to so-called counter norms, being secretive, being dogmatic, being career-driven, and focusing, of course, on quantity over quality. And she asked a series of questions to this group of researchers. First, tell me, as a researcher, the degree to which you subscribe to these norms of science over the so-called counter norms. And the vast majority of researchers agree that those norms of science are the ones that they subscribe to, that they want to participate in. The second question was, tell me not what you would like to do, but tell me what you actually do. And in that case, still a large majority reported that their own behaviors were more in line with those norms of science as opposed to those counter norms, although you can see that the hashed bars there is about a third or a quarter of folks kind of admitted to doing a little bit of both. And then the key question was, don't tell me what you behave, how you behave, but tell me how your peers, or tell me how about those folks in the other departments, other universities behave. And there was widespread perception that others' behaviors were in line with those counter norms of scientific behavior, being secretive, dogmatic, focusing on quantity over quality. And this really illustrates that collective action problem where advancement towards a little bit more transparency and some of these open science practices is widely perceived as a risky behavior, jumping into a sea of sharks. You'd like to practice these norms of science, but it looks like everybody else is just in it for their own selfish reasons. And that's a very challenging position to be in. And that's what motivates our existence as an organization, and what motivates the different types of strategies that we apply to address this collective action problem. And we organize ourselves through a theory of change to help address these types of problems. So at the Center for Open Science, we focus a lot on building infrastructure and tools to enable the types of practices we want to see happen. But more importantly, almost, are making sure that these behaviors seem more normal, that there are peers that are doing it to show that the practices are gaining widespread adoption, and that there are specific rewards in getting published, funded, and hired and promoted. And then eventually, these practices should show up in policy requirements. Today, I'm just going to have a little bit of time, and I'm going to focus a little bit on some of the examples from the Focus Ultrasound Foundation, of course, on some of the, and we'll hear more and more about those, of course, later on. But I'll be connecting some of the platform and infrastructure tools available that help support some of the policies that will be talked about in more detail in a few moments. First, I just wanted to describe what our infrastructure is. The Open Science Framework, the OSF, is an online web application that is open source and free for researchers to use. And it's designed as a project management tool to connect all the different parts of the research workflow, so that the place that you deposit your data, or that you register your work, or that you disseminate your findings and preprint servers is the same connected tool that is used to conduct and manage the work. So we connect to a number of online services to make sure that tools that are frequently used in scholarly communication are embedded in the infrastructure that we support and maintain. And the main open science practices that we advocate for and that we try to promote: study pre-registration, sharing of materials, code, and data, that those are embedded in the tools that are available on the OSF platform. And finally, I'll be mentioning how Focus Archive and collections are available to help support all of these different practices. So I'm not going to go into details about this, because more details about the Focus Ultrasound policies will be coming in a few minutes, but I just wanted to make sure that it's connected to what's going on. So I'll specifically be mentioning how the tools available on the OSF support depositing preprints, depositing data, and project management. And in a few minutes, I'll be getting to some of the encouraged and recommended practices on this policy document. But first, I'll be talking about the current requirements of the Focus Ultrasound Foundation. So Focus Archive is the preprint platform free for researchers to use to disseminate early findings of work. The purpose of preprints, of course, is to disseminate findings quickly, to get feedback from these, and to enable unrestricted access to information. An early version of a preprint is the quickest way to disseminate findings, qualified with the fact that it's not peer-reviewed yet. And we encourage updating of the preprints so that they can reflect the any errors or any updates that occur throughout the project lifecycle as reviews and suggestions come in. Those are easy to update. Submitting a preprint to the Focus Archive preprint server is as easy as clicking a button. So submit the preprint, and it takes you to a page where you can upload the PDF or the Word document. If you are already using OSF, you can select a file from the project that you already have maintained on the Open Science Framework. And then you'll be asked to assert a couple of items, whether or not data are available, whether or not the work was registered in advance. I'll be getting into more details about that. And then some more information about who you are and if you have any conflict of interest to declare. These assertions are one of the ways that we encourage and incentivize more open science practices like data sharing and registration. All of the specifics about how to do it, how to update it, how to change any information that's available on the preprint server are available on our help docs, help.osf.io. And this is just some of the information about how to create, manage, or submit preprints onto that platform. Next are OSF collections. This is a very similar page, but instead of sharing your early findings in the form of a manuscript, any materials that are appropriate to share or that you're ready and able to share can be put on an OSF collection page. So this could be data, this could be research materials, this could be analytical code, this could be early research plans, pretty much anything that you want to submit to a collection, anything that can be uploaded digitally can be disseminated through a collection and the Focus Ultrasound Foundation's OSF collection. To do so, select the Add to a Collection button on the page. I'll provide links to all of these in a few moments. And you start selecting what you want to share. Now, the one caveat here is that the items to share are already on OSF. So here's an example of OSF project with lots of materials available to share. So you select a project that you already have. And this is one of our co-presenters OSF project that just has some examples of what type of data materials are able to share on an OSF project. So, and you can see in the description of that project, it's included in the Focus Ultrasound Foundation's collection. It also has tools for creating DOIs for persistent identification, licensing it, so that you can declare how others can or should use or reuse that information that you're sharing publicly online. And again, all the information for questions and how to use or modify those are available on help.osf.io. Next, I'm going to get to the encouraged actions. And again, more details about what precisely the policies include will be given in a few moments, but I just wanted to give an overview of what these encouraged actions are for register reports and how to register research in preclinical settings before one is ready for clinical trials, how and why that process is recommended. So I'll be focusing on these two recommended practices that the Focus Ultrasound Foundation includes in their encouraged actions. In the preclinical setting, pre-registration is designed to create a timestamped read-only version of the research plan. So they will always include a study plan, the proposed research questions, hypotheses, and what type of information is going to be collected. And we strongly encourage that they also include an analysis plan, how those specific hypotheses will precisely be tested, sometimes even including analytical code. That analysis plan component can help address changes to the research plan that might, that could potentially invalidate some of the statistical tools that are frequently used. I'll get to that in a second. The main thing that pre-registration is designed to promote is a clearer distinction between planned hypothesis testing confirmatory research and unplanned discovery exploratory work. These are two equally important modes of research, but they're very easy to conflate with one another. So these two modes of research are designed to complement one another. When we are in the context of confirmation, this is very traditional hypothesis testing. The results of this work are held to the highest standards of rigor, and we want to minimize false positives. We don't want to say something is related or there's an effect between X and Y, unless we have relatively high confidence of saying so. And traditional tools of statistical inference, p-values, which we all love to hate, but those maintain their value when they're held to the strict confines of hypothesis testing. Contrast that with discovery research. This is really why we are all in research, of course. We are pushing knowledge into new areas. We really don't want to miss an unexpected discovery. You don't want to, you want to minimize the false negatives. You don't want to say something is not possible, you know, if there's a chance that there could be an interesting relationship to follow up on. However, in this mode of work, it's not about making inferences to wider populations. The probability values that we frequently use don't maintain their diagnostic value in this context. And pre-registration is really designed to make that distinction more clear for ourselves as we're going into these two modes of work. But unfortunately, when we present exploratory results using tools of confirmatory hypothesis testing, it does tend to increase the surprisingness and therefore the publishability of work, but it comes at the expense of their credibility. So that's one of the main issues to address. There are a couple different ways to incorporate pre-registration into our work. The most typical way, or the primary example, is when you're starting with a very specific testable hypothesis. So it's either coming from theory or perhaps a replication of preliminary findings. So that starts with that very detailed pre-specified pre-registration. That enables us to collect new data. You conduct the confirmatory hypothesis tests that are in the pre-registered plan. After those are conducted, one is encouraged to go into that discovery phase, look for any qualifiers, any variables that might explain differences that you didn't anticipate early on. And when those do come up, that can and should go into the next round of confirmatory hypothesis testing with new data or held off data that weren't analyzed before. Now, if we knew what we were doing, it wouldn't be called research. So this is often an ideal that's not easy to apply in all contexts. So another way to use registration in preclinical work is when we have very few predictions or expectations. We just start off designing the study as we normally do, of course, collect or access the data if it's an existing data set. And then we randomly split a portion of it and hold off a certain portion of the data. And different folks will describe different proportions of the data set that should be held off. But of course, it has to be a random subset, whether it's a quarter or half or 3/4, whatever. With the other portion of the data set, go into that normal discovery mode, look for whatever is worth taking a look at. Build and develop and test code that are using those real data. And then once something seems promising, use that to create the very specific pre-registered plan and use the held off data set to conduct those confirmatory hypothesis tests with that data that has not yet been used. When writing up the results of registered work, we want to make sure that there's a link going back to the pre-registration. All of the pre-specified tests are reported. Any unregistered analyses or anything that was not included in that plan must be transparently indicated, described as exploratory or unregistered tests, and then disclose any changes or modifications to the research plan. And that sort of goes to the subset of number 3 there. On the OSF registry, there are lots of easy tools for creating pre-registrations. They're designed for preclinical work, and they're largely designed for folks who haven't registered their own study before. So it walks us through individual step-by-step items to include in that registered plan. On the OSF registry, you can just select Add New. And as I mentioned before, you can register an OSF project that is already included on the platform if you've already been using it. But if this is your first time doing it, you will not select a registration from an existing OSF project and just go into the normal workflow of creating an OSF registration. That's the default selection on that Add New page. It walks you through about 20 questions to include in the registered plan. The analysis plan is the most detailed section of it, and it often takes, of course, the most time because that's the section where we are eager to get going and deal with some of those nuances once we get into the data set. But that's one of the most important parts of the registered plan. So I encourage you to spend a little bit of time at that stage. That's often held off until after the data are actually collected. At the conclusion of that, there is a registered page that gives you all of the information about what is included in the OSF pre-registration. A new feature on OSF is an ability to update those registered plans in order to transparently disclose changes to the plan that had to happen because of limitations with the study design or the participant pool or whatever happens. And the key there is to be to indicate when and why those changes were made. So that is a recent update that we're kind of excited to show off. Lastly, I'll mention what registered reports are in my last few moments. This is a way to guarantee publication of a registered research plan. We do expect, and we do see in the literature, that registered research will often lead to lower effect sizes or higher prevalence of null results. That is a way to address publication bias, of course, to make sure all the evidence gets out there. But when faced with negative results, we might be less likely to submit them for publication. Reviewers or editors may think that they're not as worthy as for publication. So registered reports are a way to address those concerns before results are known and unbias the literature. So it separates peer review into two stages. Once that pre-registration is almost ready to be locked down, that is what's submitted to the journal. And that first stage of peer review addresses whether or not the reviewers and editors think that the hypotheses are important and well-founded. Is the study well-powered? And are there quality control checks to ensure that any null results remain interpretable? If everyone agrees that that's true, they can be given in principle acceptance, a promise to publish the results regardless of the main outcome. And finally, once the study is conducted, the only things that are assessed in that second stage of peer review are whether or not the positive control succeeded and are the conclusions justified by the data. But very importantly, not whether the hypotheses were supported or significant or novel or impactful. There is a large number of journals that offer this, and those are available on the Center for Open Science website. With that, I would like to cede my time back to the moderators, and thank you all for your participation and attention. Have a great day.
Awesome, thank you, David. That was great and very comprehensive. Thank you a lot. Again, just a reminder that we will have time for questions at the end, so please post them in the Q&A box if you have them. And next up, we have a brief presentation from myself on the Foundation's open science policies. Hi all. I'm going to give a brief overview now of the Foundation's open science policies as they pertain to the research we fund and how it is published. I'll also discuss new policies that are coming in 2023. Our existing requirements for funded research are thus. Firstly, all research resulting from Foundation awards must be made publicly and freely available. This means publishing in an open access or hybrid journal. The Foundation is glad to support this practice by reimbursing all associated publication fees entirely. Secondly, a final report of all Foundation-funded research must be deposited in our preprint server, Focus Archive, for publication. The site is freely accessible to all. And finally, the Foundation reserves the right to ask funded researchers to deposit their study data in public repositories or to allow us to deposit it there ourselves. We have a Foundation-branded general data repository which can be used for this, as well as a specific immuno-oncology one in which we require that certain immuno-oncology data be deposited. Both are hosted on the Center for Open Science platform. So moving on, following practices, while not yet requirements, are actions the Foundation strongly encourages researchers whose work it funds to engage in. It is our expectation that researchers make the data, code, and materials involved in their research publicly available whenever legally and ethically possible. These artifacts and resources are often very central to the research that was conducted. Their availability is thus vital to both understanding the work that was done and reproducing or building on it. While not currently mandated, the Foundation is looking to make such transparency a requirement of funding in the future. The Foundation also expects the research it funds to be reported transparently and clearly. We thus ask researchers to consult standards from the Equator Network and use those most appropriate to report their research. Pre-registration of preclinical and technical research is another practice we encourage. When possible, we ask researchers to pre-register their study and analysis plans prior to conducting the work. We feel this practice resonates with the core tenets of sound scientific research by recording scientific intentions and plans in a publicly accessible space. Pre-registration both increases general insight into a given research endeavor and buoys accountability and research integrity. We do understand that this practice is relatively new to our research community and appreciate that researchers may not start pre-registering overnight. And this actually brings me to our new policies. So beginning in 2023, the Foundation will be rolling out 2 new open science policies. The first of which is that we will begin financially incentivizing researchers to pre-register their research. This will kick off with a trial program, details of which will be announced at our biannual symposium in October. And the second policy coming in 2023 is that we will begin requiring that all foundation-funded research is published with a data availability statement. These data availability statements will disclose A, whether or not data is publicly available for a given piece of research, and B, if so, where the data can be found. Thank you all for your time. You can find more information on the Foundation's Open Science page on our website. We're excited to be moving forward with our Open Science initiative and hope others in our research ecosystem will share in our desire for a more open, accessible, and interoperable status quo and help us in pushing towards it. Thank you.
Thank you, Charlie. We will now transition to presentations about tools and platforms available to put open science policies into action, some of which David touched on. And the first presentation will be given by Holly Smith and John McDunn from Project DataSphere. Holly is the Director of Technology at CORT and leads Project DataSphere's data sharing platform and collaborative research space. And John is the Executive Director of the External Control Arm Program and Business Development at Project DataSphere. John and Holly will present on Project DataSphere and how it enables open science practices.
Thank you for inviting us to speak here today. I'm John McDunn, Executive Director of Research and Development at Project DataSphere. It's an honor to be here and to connect our organizations. Project DataSphere is a nonprofit oncology research and technology solutions provider, and our mission is to improve outcomes for cancer patients by openly sharing data, convening world-class experts, and collaborating across industry and regulators to catalyze new scientific insights that accelerate delivery of effective treatments to patients. Our core principles are to honor patient contributions to science, and we believe the best way to do this is to make access to de-identified individual patient-level data available from clinical research and especially clinical trials, and that this will enable advancements in research through our open access and free-to-use platform that Holly's going to be speaking about in a minute. We partner very widely across institutions, and we have a public-private partnership with the FDA. And the goal of all of this is to accelerate evidence generation for new therapies and discoveries within oncology. And I think that one of our key differentiating features is that we really are a neutral platform to provide that data for the research environment. We're supported by the SAS Institute, and our deep ties with that institution are quite beneficial in a number of ways. And I think you'll see some of that as Holly walks through how our platform operates. As I mentioned, we collaborate extensively, building and managing multi-stakeholder groups to get the work done. Some of these groups have representatives from industry, academic institutions, multi-academic consortia, and like I said, we've got representatives from FDA and other government agencies who engage at an academic level with the projects that we have ongoing. On top of this data sharing platform that we operate, we have a number of projects. These research initiatives are really clustered in a handful of areas. One area we call Images and Algorithms is really a radiomics effort looking at automating some aspects of the RECIST criteria for solid tumor progression. We have a pharmacovigilance program looking at immune-related adverse events for those rare events that happen in patients on immunotherapies. We have a natural history registries program in rare skin malignancies, non-melanoma skin cancers. We have a under development program in pediatric oncology, looking at some aspects of emerging therapeutic modalities like cellular therapy, CAR T, and other therapies in pediatric malignancies. And then we've got a program we used to call our external controls program, which is really about innovative trial designs. And I'm just going to take 1 minute to speak a little bit more in depth about one of the instances of that research program. So one of the underlying theme of all these programs is really about accelerating the delivery of evidence for new medicines and new patient care paradigms, and improving the quality of the data that we use to generate that evidence. We've looked at this in clinical trial designs themselves, and we think that there are some really interesting ways to design flexible clinical trials that incorporate previous clinical trial experience as well as real-world data to generate evidence more efficiently. And we focused a lot of this effort in evidence generation in glioblastoma. As you may know, glioblastoma is the most common and lethal brain tumor in adults, and very little progress has been made in the treatment of glioblastoma over the last 10 or 15 years. There continue to be a number of promising therapies in early phase development, from CAR T therapy to targeted agents to the really thoughtful use of focused ultrasound, which is why I wanted to bring this to y'all's attention today. And one of the things that happens when you have a rare cancer with a lot of promising therapies is that you have a bottleneck in terms of the number of patients that are available to accrue to trials. And to address this need for more patient-efficient trials, we've been working with the analytics and regulatory science groups at Dana-Farber and at Duke University to develop innovative trial designs, such as an adaptive platform trial, the GBM Insight trial shown here. Here, patients with newly diagnosed glioblastoma have their tumors phenotyped, and then those patients are randomized onto one of the arms of the trial. But as the trial progresses and new evidence is generated for each patient and how they are progressing or responding to their therapy, those randomization or allocation proportions get changed. Using that progression-free survival, you can see that feedback arrow from the right side back into the randomization bin. And this allows evidence to be, as it's collected during the performance of the trial, to be folded back in to inform how patients are allocated across the trial, which will allow closing off arms for futility, identifying those arms that may be more effective and should be promoted to have a full-fledged phase 2 trial, and also the protocol was written in a flexible manner that allows new investigational arms to be added. To support work like this, we also developed ways to take data from completed clinical trials, as well as patient data from electronic health records, and build an external or supplemental control population of patients that can be used to support the internal randomized control arm of the trial. And this is a neat method to basically say that the experience that we've had over the last decade plus of treating patients with the same care paradigm is a useful and comparative baseline to judge the efficacy of new treatment concepts or treatment regimens. So with the two of these pieces together, we feel that we've built a flexible method for generating evidence efficiently in clinical development. And this is something that we believe can be adapted to other cancers and not just necessarily rare malignancies. So with that, I'd like to thank you all for the moment to introduce who Project DataSphere is, and I'm pleased to introduce and hand this over now to my colleague, Holly Smith, the Director of Data Sharing at Project DataSphere. Thank you.
Thank you, John, for that introduction. We're so excited to participate in today's event. Project DataSphere was started in 2014, and through our partnership with SAS Institute, a number of pharmaceutical organizations and clinical institutions, we launched our very first Dream Challenge in prostate cancer. So open science and direct access has been core to our mission, and we see ourselves as a key player in this learning health system. So we've continued to build both in volume and in breadth the types of data available within our environment, and we're looking forward to a future where we contain more data for research. So I'm going to give you just a few slides introduction and then jump right into a demonstration of our environment. Today, we've got over 209 de-identified patient-level data sets, which represents over 260,000 patient lives. We recently crossed over 140 peer-reviewed journal publications. So this is an important metric for us to really measure the value of making these data available to the general public. So it's continued to be amazing to see the work that's being conducted based on data made available in our environment. Historically, our platform has been made up of randomized clinical trial data. In the last year and a half, we've expanded to accommodate real-world data within our application, and we anticipate with the advanced research initiatives by PDS and our sponsors, we'll continue to have these data collections. So we'll continue to expand hosting curations and some customized and different use cases beyond our CT data. We've also spent a lot of time building out the restricted access use cases within our environment. So now we can host and manage closed data collaborations and research initiatives, which is exciting. And you'll see as you log into our environment, you have access to request access of NCI published trial data. Then SAS has also made a number of tools available for direct access, including their visual data mining and machine learning applications and their life sciences analytics framework. So I'll give you a bit of an intro to both of those. Here's a list of all of our data providers today. You can see it's a mix of pharma and research partners. And we're accepting curations, of course, by our user community. Our user community is global, and we've got over 3,000 active users today with around 1,500 from academia. So I'm going to jump right into a demo. When you first go to projectdatasphere.org website, you'll see a link to our data sharing platform at the header at the top. I'd suggest launching that and then saving that as a bookmark. That's the easiest way to kind of get here when you return. And before you're even logged in, you have access to some information so you can get started. So when you click on Access Data, you're able to navigate studies that are made available with our environment. You're just not able to download anything directly and or launch the analytics space directly. So keep that in mind. You do have visibility to see what's included within this repository before you request access. And then within the resources section, you have access to all of these getting started guides, including how-tos from the SAS for their analytics workspaces. And we, Project DataSphere, will publish information that we see from public resources when they've leveraged our data. So it's a decent place to come and find out some more information on how people have used these data previously. So when you request access, it takes between 24 and 48 hours for approval. And now when you're navigating, I'm going to filter down to Project DataSphere, so non-NCI data to start with, so you can see the actual files and data collections. And now when you're logged in, you're able to expand these details and view the information provisioned by a data provider, but then also some of the metadata from clinicaltrials.gov. So we're connected to their API and bring that information along with us. And then you can launch 2 ClinTrials directly from our application as well. So if you want to find some further information about these published studies, you're free to do so. And then you can also expand the show files, and here's where you can directly download these contributions to your environment. So in order to leverage the analytics space and leverage these data within SAS, you would use this right-hand pane and click on BDMML or LSAF. Log into the analytics space. You need to remember this username, this NPM, and then followed by a series of numbers and letters. That's what you'll use to log into the SAS system as well. And then just use your same password. So you'll see that this navigation search bar on the left-hand side allows you to down select certain areas of interest. And one area may be cancer area. So if you want to view which studies are part of this repository, say within lung, you could down select to lung and kind of just view these. If you also wanted to only look at Project Datasphere contributions, so non-NCI studies, you can down select to those PDS studies. So these you would all have instant access to for analyses. And then one other area to point out, within this linked data section, you'll see the Prostate Dream Challenge contributions are here, the relationship between the base studies and the curated set, and the work that RTI International conducted in enriching some of our base trial data. Same thing, you can see the underlying study and then the curation. So to evaluate and run analyses, you would really want to leverage both. And if you were interested in requesting access to the NCI studies made available here, I'm going to just show you an example at the top. You can see with my user login, I've got this active link that says added to NCI data request. And then the second study made available here, it says request NCI data access. So you can request up to 5 studies via one time. And as soon as those are added to, say, my shopping basket, I can view these requests underneath your email address, your account information. So they show up under my data requests. And then once you have all the studies available that you're interested in gaining access to via NCI, you would go ahead and complete a request form. Now, to get started in the analytics space, there are kind of 2 different workspaces that you have access to. The one, Life Sciences Analytics Framework, is a good base SAS application for you to do analyses and to share analyses with other users. So when you launch LSAF, you log in with your username, and you'll see this navigation pane on the left. So within your repository view, under SAS and Files, there's this PDS folder. This includes all of the clinical trial studies referencing the PDS UID. So this underscore and 3 digits is our typical format for a unique identifier within our environment. And we've also made, Project Datasphere has made some base analyses conducted against some of these trial data available for review and or use. And then the sandbox area is a space where you can publish analyses and share that with other users. The other thing, if you do have access to NCI studies, you'll see a folder in here with NCI and a number of those trials that you requested access for. So then when you launch visual data mining and machine learning, or the VDMML application, you'll see that this is a pretty robust software solution. So we've got full base SAS, of course, and a lot of data preparation modules, and then visual analytics. So this explore and visualize data, and then the machine learning modules that are part of this entire software package. One thing that John and I worked on this last year was building out a bit of a proof of concept for external controls project within pancreatic cancer across a number of different trials contributed within our environment. So this was a base level visualization to explore these studies, both comparatively, for patient population and makeup, but then also treatment performance, and to offer some base understanding as to whether or not they're appropriate for the inclusion criteria for external controls. So this base view is a study comparison, so you can easily see across these 4 sponsor trials, both volume of data, some of the demographic summary information, and then overall survival data. And then we have it broken down by sponsor, which is kind of nice. You can look independently, see both these demographic information and survival and ARM details. And then part of the data preparation and transparency for leveraging these data for external controls or supplemental controls is some evaluation of our lab information and harmonization of the units. So offering some perspective around which units were different and which ones were actually harmonized. And then a really nice capability as far as dynamically being able to display, these lab results given some criteria for patient populations. So this was a nice exploration basis set, leveraging SAS visual analytics based on data made available within our environment. And if you've got other use cases or are interested in kind of getting started here, please reach out to us, someone from either Project DataSphere or from SAS Institute. We can give you the right information and the best way, basically, to get moving. So thanks so much. That was all that I had to present. I hope that you all enjoy the rest of the session, and we'll see you soon.
Thank you, John and Oli, for this presentation. We wanted to mention that we also have two pre-recorded tutorials on how to use our Focus Archive and how to use the FUSF Foundation data repository. We decided not to play them live today to have time for question and discussion, but they will be included in the recorded version of the webinar, and they will be available online. And so now we really have time for question and discussion. So if you have any questions, please use the Q&A tab on your Zoom page. And we'll start with the first question, so to Holly and David, which is really about how to incentivize our community, the researcher, the clinicians, and the companies, especially, for example, when companies are sponsoring clinical trials. What can we do? How can we incentivize them to really use and follow these open policy practices? Is there any special incentivize that we can play with, except with the contracting, for example?
Holly, would you like to go first?
Sure. From Project DataSphere's perspective, we've been working both closely with FDA. Last year, we hosted a seminar all around the topic of data sharing, and it's a tough topic, right? So everybody wants to do it, but then when we actually look at the active participants, it comes with a lot of conditions, I guess. So the incentive piece is a little bit of a tough nut to crack without having real policies in place, right? So what we've discovered within Project DataSphere, we can incentivize sharing through focused research initiatives. There's kind of this one avenue where people feel comfortable in wanting to make data available for research collaborations, right? Another voice that I'd say is kind of a key piece here is really from the patient's perspective. So I think patient advocacy groups kind of play an important role when we're working together to identify good sources of information to bring together data collections, I'd say. And then third area is really focusing and publicizing and I think enabling all of us to communicate kind of around the same terms, language, and processes that are in place. I know NIH has been working really hard on their new data sharing policy. I think that's going to be enacted the start of January 2023, but they've already got a lot of resources kind of out there in the public space. So I really encourage everybody, if we can start to leverage what's acceptable when it comes to data sharing forms, templates, data use agreements. So integrating some of that acceptable language, making it a little bit easier. So whatever we can kind of do collectively to remove some of these barriers to entry, and then really start thinking about sharing practices early on and trial planning. So when we think of the entire data life cycle, this ask for data comes sadly too late, I think, in the discussion. So I would say it really should be core to trial plans, and we should be accommodating and making sure that there are resources and there are standard approaches, for both formatting of these data and a sharing policy that feels comfortable, for where we're at in the study. So those are just a few areas I think about.
Yeah, and I would absolutely echo everything that Holly said. Especially thinking about asking for a data set at the end or following up with those data available upon request type of mentalities it's often too late. That the data are somewhere else out of mind, and I'm just focused on getting my publication out the door, or of course, the next 2 or 3 studies on the plate. So making sure that becomes part of the study planning process. From our perspective, we often think about all of the different nudges and incentives and policies that individuals encounter on a frequent basis. So getting funded, of course, Focus Ultrasound Foundation policies, getting published, and traditional academic rewards and hiring and promotion. Those all need to include recognition for the time and effort it takes and contributions that are provided through data transparency. And then there are a lot of those kind of, I'll sound a little bit judgy, but essentially selfish reasons for a lot of these practices. They make one's work more efficient. There's a learning curve, there's a barrier to getting over that. But spending that time and investing on reproducible practices makes one's own internal research processes much more efficient, makes reusing one's own data sets a lot easier for future work. And then, very basic things like citation counts are higher when there's an association between openly available data sets and preprints and activities like that, and getting cited more frequently. So sharing evidence like that is important as well.
And about this training phase that you're mentioning on how to learn these practices, is there a standard, I don't know, training to follow? Is there something that you would recommend to say, do that, follow that, read that?
Yeah, one of the projects that we have is called Overcoming the Knowledge Barrier. And it's really about taking the first step in any of these practices, but particularly a lot of the reproducible data analysis and data sharing policies or practices. A lot of that can be quite difficult to learn, but there's a lot of resources available, largely online these days, of course, for learning these. And if I can, I'll make sure that gets shared after the webinar.
Thank you.
Yeah, fantastic. And David, I actually had a question that was more just for you. So we know the Center for Open Science works in many different research fields. I was wondering if you could talk a bit about how a medical research field compares to kind of other fields in terms of average open science kind of standards, practices, behaviors, that kind of thing.
Yeah, that's a great question. And that's something we've been pondering a lot recently. One of the tools we've been using is the Open Scholarship Survey, OSS, to evaluate the perceptions and practices and attitudes of a variety of open science practices across disciplines. So in the medical research community and a lot of folks who work at the Center for Open Science or a lot of the open science community are psychologists. We have a lot of efforts with education research communities and several others. And comparing how attitudes vary across disciplines is important for how we engage policymakers and stakeholders in each discipline. There is some difference here and there. The same pattern tends to hold up very frequently, where a lot of these types of practices are attitudes are very favorable towards them, but adoption and perception that these are a little bit risky is pretty widespread. I would say a lot of the attitudes about reproducible research practices are pretty favorable in the psychological science community, and a lot of practices have been taken up there, especially in the preclinical world. Attitudes about data sharing are very iffy in education research because of the multiple levels of informed consent and ethics concerns with teachers, parents, and administrative bodies. In the medical research community, there's a lot of very conservative focus on intellectual property. And we've been working with a variety of IP lawyers at universities to discuss how pre-registration can help support IP claims because those are citable assertions of a project going forward. Many of those are embargoed until a maximum of 4 years, and that helps with a lot of the questions that arise that are distinct from one community to the next.
Interesting, thank you. That's a great answer.
And a question maybe more for Holly about then what resources are required to really start an open science practices or data sharing. For example, for our community, platforms are available like the Project Datasphere platform, but is data curation, for example, a big effort to in order for the data to be available and mineable for everybody? Are bioinformaticians, for example, really required to be part of the team in order to conduct meaningful analysis? So what's let's say for a group who wants to start, or for a foundation like us to want to push this effort, what resources do we need, or do you think we need?
Yeah, I would say at a basic level, of course, familiarity with clinical trial data capture design. So within our environment, we do share if those data are conforming to, say, CDISC standards, as a potential starting position. But I would say that every provider and sponsor for these studies, there are some differences across. So having a resource that's both familiar with those formatting and depending on what your analysis is or problem statement, you will likely have to do some harmonization across. So there are some studies that PDS has kind of made available in an analytics space, so that may be a good place to get going. But I would say that would be your initial work is looking across to make sure that the studies have the endpoints that are of interest or the data elements that are of interest. And then there will be some work for harmonization if you're leveraging multiple trial data, for sure.
Thank you.
Yeah, thank you very much. I appreciate it. I think we're almost out of time, and so I think we'll wrap it up. A big thank you to our speakers, David, Holly, and John.
Thanks so much for inviting us.
If you have questions or would like further information, please visit us at fusfoundation.org or email us at info@fusfoundation.org. And stay tuned to our newsletter and website for invitations to future webinars. And Fred, do you want to say a few words about the next installment?
Yeah, just the teaser, we are also planning another installment of these discussions with more focus on technical implementations and very more focus on our immuno-oncology community to identify our technical needs and set up course of action to create these data repositories and bioinformatic tools, starting certainly with a preclinical flow cytometry data. So stay tuned, and we'll be communicating shortly. Thank you very much.